Hi everyone, this is the Bio 285 final exam review, part 2, and we're going to go ahead and get started. Just a few important reminders that this review session today is only 25% of what you should know for the final. There are 4 review sessions total. There was one yesterday, there's this one today, and then there's 2 more that are left. So there's one on Monday and I believe on Tuesday. So make sure that you at least know that this isn't the only information you'll have to know for the final. And then another thing I wanted to say was try not to just use only these review sessions as a mechanism to study for the final, because we only included the most important things from each week or things that we thought were super important, but there definitely are other details that we did not include and that we highly recommend that you go back into your notes and review. And yeah, and the final, I believe, is on Wednesday, November 2nd, and it opens at 3 p.m. Someone let me know in the chat if that sounds correct. That's what the...
I think it's December 2nd.
December? What if so sorry. I wrote November. I'm sorry. December 2nd. Thank you.
No problem.
At 3 p.m. So yeah, those are just reminders, and we're going to go ahead and proceed with the 55 stuff. Sebastian, do you want to go ahead?
Oh, yeah. Sorry. Okay. Gene expression and regulation. Could you skip to the next slide, please? Okay, so just very quickly, here's your central dogma. Central dogma, you guys probably know this, so I won't go like ham on this, but essentially you obviously have DNA. DNA is composed of a bunch of genes that code for some kind of protein. So when your cell needs some protein, like a channel or whatever, it's going to send a polymerase and transcription factors to the DNA. It's going to make a copy of the gene for the specific protein that it needs. And then that copy, which is the mRNA, is going to be translated into that protein, that channel, for instance. Okay, you could go on. Okay, so here's some ways that process is controlled. So like I said before, if the cell needs something like a channel, it's going to send DNA polymerase and transcription factors. That's one way. I'm going in like opposite order, but whatever. That's one way that transcription is regulated. To actually get your gene transcribed and ultimately translated, you of course need transcription factors, or you need a DNA polymerase, but with the polymerase, you also need transcription factors. So the polymerase can't really do its thing without the transcription factors. And those transcription factors can either promote transcription or they could suppress it. So again, that's just one way that transcription is regulated in the cell via DNA polymerase and its transcription factors. Another way that it's regulated is through chromatin compacting. So like I said, your DNA has the genes for all kinds of proteins that it could need. But if there's a region of DNA, if there's a stretch of DNA that is not needed for whatever the cell needs, it's going to compact itself. It does this by just wrapping itself tightly around like a histone, which is like this protein ball thing. And because the DNA is tightly woven around this histone, it's not allowing the polymerase or transcription factors to attach itself there. However, if there is a gene that the cell does need, then the chromatin or the DNA would like loosen its grip around the histone. That way, now that the DNA is loose and not so tightly woven around the histone ball, the polymerase and the transcription factors can access that part of the DNA and transcribe whatever gene it needs to transcribe. Okay, you can go on. Go on. Okay, so sigma factor 70 is a transcription factor. Like I said before, a transcription factor is just something that helps the DNA polymerase bind onto the DNA, and it also does a promotion or a suppression action for the DNA polymerase. Sigma 70 is a transcription factor. It's unique to bacteria. I'm not going to go through like every single detail on this slide because of course this will be accessible to you during the exam. So In the chance, I doubt it, but in the chance that there's a Sigma 70 question, you could just refer back to the slide. It has like a summary of pretty good, it's a good summary of all things you need to know about Sigma 70, but just the basic overview is that it's a transcription factor that's unique to bacteria. Okay, you could go on.
Also, I just wanted to interject real quickly. If people have questions at this point, you can feel free to put them in the chat or unmute yourself to ask a question. But yeah,
I've actually just got one quick question.
Yeah.
Was review 1 ever added to the TA YouTube channel?
Yes, I think so. Did you click the link for the YouTube in the email I sent yesterday? Did it not work?
I don't think it worked for me.
Okay. I will look into that while we're going through here and make sure it's updated. Sorry about that.
That's okay. Thank you.
Mm-hmm.
Yeah. Okay. Thanks, Michelle. Okay. Yeah, and of course, just to reiterate what Michelle was saying, if you guys have any questions at all, of course, feel free to interrupt me or drop something in the chat. What am I looking at? Okay, transcription factors. Okay, so this is, again, just another summary of what I said before. The RNA or the DNA polymerase can't bind onto the DNA on its own. It needs something to like guide it there, to help it bind itself there. And again, those are transcription factors. So they can either promote transcription, or they could suppress it. The promoter or the promotion transcription factors are called positive transcription factors, and the other ones are called negative. Those are the ones that suppress transcription. Like I mentioned before, Sigma 70 is one that's unique to bacteria like prokaryotes. Eukaryotes, like our cells, we don't have Sigma 70. We have these things called basal transcription factors. They work in very similar ways, slight differences, but again, it's not like a huge, you don't need to like go into crazy detail about those. But just know that Sigma factors, like Sigma factor 70, that's unique to bacteria, whereas we have basal transcription factors. Any questions about this? Okay, go on. Okay, so I talked a whole lot about transcription now, or excuse me, like the steps needed to get to transcription. Let's talk about how transcription actually works. So in eukaryotes, we of course have exons, which are the parts that will ultimately code for the protein, but surrounding our exons, we have a bunch of introns. Those introns need to be spliced out before the gene can get translated. So RNA processing is just the process of making mRNA, which, remember, this process is unique to eukaryotes because bacteria, they don't have introns, or they don't have introns because they don't have this RNA processing control. Bacteria transcribe and translate at the same time. So they don't have introns because they don't have any way of splicing those out. But eukaryotes are definitely much more complicated mechanistically than bacteria. So we do have introns, but those obviously need to get spliced out. We do have the mechanisms to slice those out. We do have the proteins to do those. They're called spliceosomes. And essentially spliceosomes are just proteins that help create the mature mRNA. So just to reiterate everything that I've said so far, the DNA polymerase can't bind to the DNA on its own. It needs help with transcription factors. Once the transcription factor binds the polymerase to the DNA polymerase is going to start polymerizing. It's going to make its transcript. Once it makes the transcript, the spliceosome has to come in, cut out the introns. You have to attach your 5' guanine cap, your 3' poly A tail, and there you have your mature mRNA transcript ready to find a ribosome and become a protein. Okay, any questions about that? And again, I just want to reiterate that this process, the RNA processing control, it's unique to eukaryotes. Prokaryotes don't have processing control. That's a very, I don't want to say common, but like it's a very easy thing to quiz you on, to test you on. So definitely just make that differentiation in your head. Okay, go on. Okay, this is a little more about what I talked about before. Wait, what is this? Oh, okay, yeah, this is just the process of making the mRNA transcript. Like I said before, your DNA polymerizes the specific gene that it needs for a protein. Then you're processing that transcript involves cutting out the introns, attaching the 5' cap, the poly A tail at the 3' ends, and there you have your mature mRNA transcript. Again, I'm kind of glossing over a lot of like the details here. I don't just want to like read off this slide, obviously. But if you do have any questions about anything, feel free to let me know. Of course, these slides will be accessible to you during the exam. So hence also why I'm not going like in crazy detail about all of these stuff. But again, of course, if you have questions, feel free to ask. Okay, go on. Ugh, it's hot. Okay, so everything that I talked about before with the mRNA, intron splicing, and the 5' cap, whatever, that is all unique to eukaryotes. Now we're going to talk a little more about prokaryotes. So when you're going through questions like this, or when you're going through this chapter, I guess during your final, please be sure that you know the differences between these. We actually have a slide that has a very nice summary of the differences. So again, that's another way, or that's another slide accessible to you for you to make those differentiations. You don't have to like go crazy trying to memorize all of these different things. But anyway, gene regulation in prokaryotes works a little differently. Prokaryotes have these things called operons. Operons, it's not like an assembly line, but it's kind of like an assembly line. Essentially, you have your promoter region. It has the operator region, which essentially tells the polymerase, like, can you transcribe, can you not transcribe? And then it has its structural genes. Most of them are polycistronic, meaning that one operon will code for multiple genes. There's also monocistronic, which means that one operon is going to code for only one gene. We're mainly going to be talking about polycistronic operons, so operons that code for multiple structural genes. But this is essentially what it is. So again, it's kind of like an assembly line. You have your promoter region, which is where the polymerase binds to, the operator region, which tells the promoter start synthesizing or don't start synthesizing, and then your structural gene parts, which are just the genes that the operon is responsible for. Okay, any questions about this? Okay, go on. Okay, here's that slide I was talking about with the different differentiations. Again, it's going to be super useful for you to bear in mind some of the differences between eukaryotes and prokaryotes. When you're going through questions like this on the final, please be sure to know the differences. Sorry, let me just check out the chat real quick. I'm a little confused about the gene regulatory versus the operator. The regulatory gene. Okay, could you go back to the previous slide, Michelle, please? The gene, the regulatory gene. Hey, Sophia, do you mind like clarifying for me which part? Oh, okay, the regulatory gene. Okay, I see what you're saying. What did that stretch code for again? I honestly don't remember. Do you guys remember, Michelle or Saeed? Right there with where that yellow stretch is?
That goes for TRPR, right? Yeah.
Is it TRPR? It might be TRPR, regulatory gene. Yeah, so all of your diagrams are going to look a little different. I think the regulatory gene part, like that stretch before the promoter, I think that's just the region where a repressor is synthesized. So again, operons, you're going to find like 1,000 different diagrams of an operon, like on Google Images or something. I'm pretty sure what that stretch is, that little yellow stretch right before the promoter region that says regulatory gene, I think that's just a gene that codes for a repressor.
Okay.
Sorry?
Okay, yeah, that makes way more sense. I was just
okay.
confused what the purpose was between the I and the O, but that makes a lot more sense.
Yeah, no problem. does the repressor usually bind to the operator region to stop transcription? Yes. So, well, not to prevent transcription from happening. So, Julia, to answer your question, that regulatory gene region that codes for a repressor, that repressor binds to the operator region. So, if the repressor is bound, transcription will not happen. It cannot happen because there's a repressor sitting on the operator and the polymerase can't move. If transcription really does need to happen, then the repressor obviously won't be bound to the operator region, and it'll allow the polymerase to do its thing. Sweet. Okay. And again, here's your slide with all the differences. Okay, my favorite. Trip operon. Okay, yeah, so like I said, all of these different operons are going to look slightly different. But essentially here at the purple region, or all the way at the far left, that's your TRPR repressor gene. So again, that gene just codes for the repressor, which will bind to your operator. Right there, okay, awesome. The diagram tells you it's where the repressor binds. Okay, one thing that I do want to clarify, this actually came up on the first exam, so most of you probably know this already, but just to reiterate, the TRPR region, that itself is a gene, which means that it has its own promoter region. So there was a question on the first exam that said something like, the TRPR repressor gene's promoter region was mutated and the polymerase can't bind there. What I noticed was that a lot of people mistook that as the promoter region for the structural gene parts, for the TRPE, TRPD, whatever. So just to clarify, again, I know you guys probably know this already, but just to clarify, the TRPR gene is a gene that codes for the repressor, which means that it has its own promoter region. That's the promoter region that was mutated in Laura's question. However, the promoter region for the structural genes, which is where you see that P, right before the O for the operator stretch, that P is the promoter region for the structural genes. So just keep that in mind in case something like this shows up again in the final. Know that each gene requires its own promoter region. Those structural genes, the TRPE, TRPD, whatever, again, this is the polycistronic stretch. So all of those are going to be transcribed with the promoter region. However, because the TRPR is not part of the structural genes, it's its own gene, it requires its own promoter region. Okay, any questions about this part? Okay, go ahead. Okay, what's the attenuator again? I will definitely talk about the attenuator in maybe a slide or two from now. First, I got to figure out what this slide is. Each operator continues. Oh, okay, yeah, so this is just, I guess this contextualizes everything that I said before. So all the way at the far left in that purple region, that P is the promoter region. That's where the DNA polymerase is going to bind to, and it's going to code for all of those structural genes. The operator is just the section that essentially tells the polymerase, can you start transcribing or can you not start transcribing? We didn't talk about the LAC operon in this class, right? Only the TRP operon? Yeah, I think we only talked about the TRP. So let's just concern ourselves with the TRP operon in this case. Okay, do you guys have any questions about this slide?
Can you repeat again what the operator is doing? I'm sorry.
Yeah, the operator, so on the right diagram, there's a red box that's highlighting that O. That's the operator region. And what the operator region does is that's where the repressor sits. If the repressor is sitting in the operator region, it's going to block the polymerase from transcribing. But if the repressor is not bound to the operator region, then the polymerase is free to start transcribing. Does that clarify your question?
Yeah, so that's where where the repressor binds. Is that also where in the TRP operon where where it would have the regions 1, 2, 3, and 4? Is that also what the operator was doing?
Oh, wait, I'm sorry. Could you repeat that last part, please?
Is that where like I remember when for the exam there was like a region like 1, 2, 3, and 4, and like
Oh.
depending on like
Yes. No, that region is called the leader region or the leader sequence. This diagram doesn't illustrate the leader sequence. It is there, like it does exist. Again, all of these diagrams are going to look slightly different. But no, that operator region is not where the leader sequence is. We will talk about the leader sequence, which has regions 1 and 2 and 3 and 4, the attenuator mechanism. We'll talk about that, I think, in the next slide, I believe. Okay, so the way the trip operon works is the cell obviously wants to conserve its energy as much as possible. So if the cell has tryptophan freely floating around, it doesn't need to synthesize more. That's just a huge waste of energy. So if tryptophan is around and it's freely diffusing around the cell, it's going to bind to the repressor. The repressor is going to bind to the operator region, and it's going to prevent the polymerase from transcribing genes that will essentially that will ultimately make tryptophan. And again, the repressor is bound when tryptophan is high because the cell already has tryptophan, so there's no need for it to make more of it. If tryptophan is low, if there's no tryptophan freely floating around, then it can't bind to the repressor gene because it's not around. So the repressor gene can't bind to the operator region. If the operator if the repressor can't bind to the operator, then the polymerase is free to start transcribing. Okay, any questions about this? Okay. Okay, you can go on. Okay, could you go on to the next slide, please? I just want to skip this one. Okay. Okay, so I mentioned before, this, by the way, everything that you're looking at right now, this stretch, to answer your last question, whoever asked the last question, this is the leader, this is the leader sequence. It comes after the operator and before the structural genes. Okay, so this makes a whole lot more sense if you consider tryptophan and tRNA tryptophan as two different things. Consider those as two different concentrations. So I mentioned before that when tryptophan is high, tryptophan is bound to the repressor is bound to the operator, and the polymerase just isn't going to bind to the DNA. If tryptophan is low, there's no tryptophan available to bind to the repressor, so the repressor is not going to bind to the operator, which means that the polymerase is free to attach itself to the DNA and start transcribing. Okay, so that's the first level of regulation. That's the tryptophan concentration and level of regulation. The tRNA tryptophan is, I don't want to say more complicated, but it's a little more involved, I guess. So again, consider these as two different concentrations. So let's look at this scenario where tryptophan is low, but tRNA tryptophan is high. So when tryptophan is low, the repressor is not bound to the operator, which means that the DNA polymerase is free to start transcribing. So it's going to start transcribing because tryptophan is low. But let's look at the situation where tRNA tryptophan is high. So your polymerase is going to start right there at the number 1 at base pair 1. And it's going to start transcribing that whole thing. It's going to start transcribing all of that until it gets to transcription pause, which is right here at base pair 90. It's a little hard. I keep saying like right here, but I don't know. You guys obviously can't see what I'm, wait, actually, could you see my cursor by any chance?
Yeah.
No? Wait, you can? No way. Oh, I think that's Michelle's cursor. Wait, okay, I'm up here. I'm touching the high on tRNA tryptophan is high. Can you see that?
I can't see your cursor, but if they can see it, then that's okay. That's fine.
Okay. I think they were just looking at your cursor, but it's okay.
Oh, okay.
Yeah, no, you're fine. Anyway, okay, so your DNA polymerase is going to start transcribing. It's going to start at 1, and it's going to go all the way, up until it gets to 90, base pair 90. That's where transcription pauses. It pauses so that the RNA, it pauses so that the ribosome can start translating what the polymerase just finished transcribing. So, okay, the ribosome is going to start at the AUG site, at the start codon, which is on base pair 27. The ribosome is going to bind right there, and it's going to start coding. Notice at base pair 54, there is a UGG region. Those UGG codons code for tryptophan. So this is where the tRNA tryptophan concentration becomes important. If tRNA tryptophan is in high concentration, the ribosome is going to place its codons there, and it's going to continue moving. It's not going to have a problem, synthesizing that stretch because there's plenty of tRNA tryptophan going. So it's going to move on from that site with no problem, all the way until it reaches the UGA region, which is the stop codon. So mind you, if the ribosome translates everything and goes straight through the 54 region and goes all the way to the 70 region, which is the stop codon, look at where it landed. It landed on region 2, let's say. So that's where the polymerase is sitting, that's where the ribosome is sitting right now. It's sitting on region 2. Okay, now the mRNA, the DNA polymerase is going to continue transcribing. When it continues transcribing, it's going to transcribe regions 3 and 4. So notice that 3 and 4 are forming a hairpin loop. Regions 2 and 3 cannot form a hairpin loop in this situation because there's a huge ribosome sitting on region 2. So because that ribosome is sitting on region 2 cannot hydrogen bond to region 3. It's occupied right now. So the only regions that can bind are regions 3 and 4. When regions 3 and 4 bind, it's going to yank the DNA right out of the DNA polymerase's reading frame, which means that the DNA polymerase is no longer attached to the DNA, so transcription stops. So this is the second level of regulation where you would get an attenuated mRNA. It's shortened because the cell has enough tRNA tryptophan, so it doesn't need to synthesize more. Any questions about this? I know this is it can be a little confusing, so of course feel free to ask if you're confused by this. Okay, you go on.
Sorry.
Ding. Okay, so I just talked about what happens when tRNA tryptophan is high. Let's talk about when it's low now. Okay, so same deal as before. The DNA polymerase is going to start at base pair 1. It's going to continue transcribing all of that up until it gets to region 90, which is that transcription pause site. Pause is there so the ribosome could catch up to it. So again, the ribosome is going to connect to base pair 27, which is where the start codon is. It's going to transcribe until it reaches region 24. So in the previous example, we had a situation where tRNA tryptophan was high, meaning that the ribosome was able to place its codons there with no problem and continue translating. However, in this situation, there is no tRNA tryptophan, or it's extremely low. So if there's no tRNA tryptophan, the ribosome can't place any codons there. It can't translate that region because there's no tRNA tryptophan available to it, which means that it's just going to pause there. So again, in the previous example, the ribosome paused at the stop codon, which is region 70, which is sitting on region 2. In this example, the ribosome is now paused at base pair 54, which is where the tRNA tryptophan codons are supposed to go. It's paused there because there are no tRNA tryptophan codons. So now the ribosome is stuck on region 1. Because the ribosome is stuck on region 1, it cannot bind to region 2. So if you remember from the previous example, region 1 was bound to region 2. But now because the ribosome is stuck on region 1, it can't bind to region 2 anymore. The regions that will bind, however, are regions 2 and 3. And the reason that 2 and 3 can bind to each other now is because the ribosome is no longer sitting on region 2 like it was in the previous example. So when you get a hairpin loop with 2 and 3 connected to each other, it will not attenuate your mRNA. You will have the full length transcript. Okay, any questions about any of this? Okay.
No questions?
they're just
So
so.
I do have a question.
Yeah.
So in the last slide, you said that the hairpin loop wasn't forming between region 1 and 2 because the ribosome was sitting over it.
In this example, in this slide,
Yeah.
the transcript is not formed. Sorry, could you go back to the last one?
Yeah.
The transcript is not binding to regions 2 and 3 because the ribosome is sitting on 2.
So then in this, is the hairpin between 2 and 1 forming, or is it not?
Yes, in this example, the hairpin is forming between 1 and 2,
Okay.
and it's forming between 3 and 4 as well.
Okay.
But in the other example, it's not. 1 and 2 is not forming.
So the ribosome is over region 2, but the hairpin is still forming?
Yes, except
Okay.
the important part is that it's not forming with 2 and 3.
Right. Okay. Gotcha. Thank you.
No problem.
I have another question.
Yeah.
Could you go over what happens at like base pair 70 again at the UGA?
Yeah, UGA is the stop codon. So that's where the ribosome is going to stop. That's where it pauses.
And so regardless of like the levels of tryptophan, it will always pause there?
No. So if tryptophan is high, if you haven't, if tRNA tryptophan, remember you have to consider tryptophan and tRNA tryptophan as two separate things. If tRNA tryptophan is high, then the ribosome will pass. Do you see region 54 that has the trip part?
Yeah, I understand that part, that it depends on the tRNA, the
Right.
trip tRNA, but then what exactly does the position 70 depend on?
Nothing really. It's just like a stop codon. So that's where the ribosome is just going to stop. I don't know if I'm like answering your question at all.
So if it doesn't specifically depend on anything, does it always stop there at some point?
Oh, okay. Yes. Ultimately, it will stop there. So in this case, tRNA tryptophan is low. So the ribosome is going to pause at section 54. But that's not to say that it's going to stay in region 54 like forever. It's only going to stay there until it finds enough tRNA tryptophan. Once the ribosome does find enough tRNA tryptophan, it will continue up until the UGA, up until the stop codon. So yes, ultimately they always do end up at region, they always do end up stopping at the stop codon. However, it just depends if your tRNA tryptophan is high or low. If it's low, it's going to pause momentarily at the region 54, at base pair 54. Does that answer your question?
Yeah, I think so. Thank you.
Okay, no problem. Okay, you can go on from this, Michelle, please.
Okay. Yeah, I just want to say I'm glad you guys are asking questions, because I just felt like for me personally, this was one of the tricky concepts from this class. So it's good that you guys are asking questions.
Yeah, hopefully there's not a lot of this on the exam, but who knows?
Okay, so hi everyone. I'm going to be going over RNA interference, or RNAi. So we have three types of different non-coding RNAs, or different types of RNAis. The first one is going to be your small interfering RNAs, or your siRNAs. And these are typically going to be used by the cell when there's going to be an outside invader, or like an exogenous source of DNA. So you're going to want to think of like bacterial or like viral infections that the cell has to fight off. So yeah, this basically is used by cells to protect protect itself from foreign invaders, and can also be used a lot in research purposes, which we'll talk about later. And then you have your microRNAs, or miRNAs, and these are going to be encoded by the genome of the cell itself. So these are going to be used for target mRNAs that are going to be coming from endogenous sources or from the cell itself. And in humans, these are going to be thought to regulate at least 60% of all of our protein coding genes. And then the third type is your long coding RNAs. And all you really need to know for these is that they're going to be involved in X chromosome inactivation. So next slide. First, we're going to be going over RNA interference, or just like more on that. It's going to be a post-transcriptional formal control for mRNA, and likely evolved to protect cells from endogenous and exogenous harm. So comparing and contrasting the two types of miRNA and siRNA, first off is that the microRNA is going to be, again, going to be finding off endogenously expressed mRNA, and then siRNA is going to be dealing with basically pieces of target mRNA that's going to be exogenously incorporated. So like basically, again, something like a viral infection. And then a good thing to know for the exam is that she'll always ask about RNAi's relevance to research purposes. And basically all you need to know for that is that it's going to be used a lot for protein knockdown mechanisms. So basically, in the lab, you're going to inject RNAi into a cell, and then basically see like what the effects of that going to be are. So you're going to see, oh, if this turns off this protein, then how's that going to affect the phenotype of the cell? So yeah, that's pretty much that for. Oh, and also you're going to want to think about problems with RNAi. So basically, one example of that could be researchers would have to figure out how to deliver the RNA safely into the cell and effectively in a way that it'll be able to match with the target mRNA that's going to be the target for the protein knockdown mechanism. So next slide. Okay, so now we're going to be going over the miRNA pathway. Basically, it starts off within the nucleus of the cell. So your DNA, or its DNA, is going to transcribe the PRI miRNA. And yeah, so there's like a mistake on the slide here. Instead of pre-mRNA, it's first going to be PRI mRNA. And then when that's transcribed, it's going to go through Drosha processing, which basically it's just going to make snippets of it that are going to be small enough to be exported out of the cell. And at this point, this is when you're going to have your pre-miRNAs. So after they're exported, then they're going to be in the cytoplasm of the cell, and where they're going to undergo dicer processing. And that basically just cuts off the hairpin loop, as you can see on the slide. It's just going to cut that off of the miRNA. So then you're just going to be left with your miRNA duplexes. So you're just going to have your double-stranded pieces of RNA, which are then going to be further processed by dicer. And basically, dicer is just going to be a complex which has an Argonaute protein that's going to be the catalytic portion of that. And that's just basically going to unwind the miRNA duplex into one guide strand and then one strand that's just going to be degraded. And it's going to use that guide strand to either have an exact match or a partial match to whatever target mRNA is going to be going for in the cell. And then at that point, one of two things can happen. Either one is the less common one, which is if it's an exact match with its miRNA, then it's just going to be cleaved by the Argonaute protein and then subsequently degraded. Or you're going to have your partial match, which is more common, which is where if a guide strand is just good enough, it doesn't have to be like an exact sequence base for base matching, but if it's generally high enough, then you're going to have either translational suppression of that target mRNA, or you're just going to have it be sequestered into P-bodies. And the reason for that is that way the cell can use it for later instances. So basically, if a cell has a piece of mRNA, or like say a protein that it doesn't need, it'll use miRNA to be able to sequester it for later use. And then if we want to go to the next slide, we can go on. We'll go over SI pathways. Yeah, so siRNA pathways, basically the main difference is that it's going to come again from an exogenous source, so you're not going to have to deal with any of the processing that miRNA has to do with in the nucleus. So basically it's going to start in the cytoplasm of the cell, where you're going to have either double-stranded RNA or shRNA, which is basically going to get processed by dicer. into, again, the same duplexes as before, which are then going to come together with your organelle protein to form the RISC complex. And then the same thing's going to happen again, where you're going to have RISC unwind the 2 strands and pick one to be the guide strand. And another difference here is that in siRNAs, they're going to be more than not exact matches, so they're just going to be marked for degradation. And that makes sense too if you think about a cell doesn't really have any use for a foreign invader it wouldn't need to sequester it. It would just basically be well off with just degrading that. And then if we go on the next slide, we just have a side-by-side comparison of the 2 pathways. So again, the really only 2 differences are your miRNA is going to have that whole portion inside of the nucleus there that you're not going to need to see in siRNA. And then the second difference is that the ending result, mostly in your miRNA, is going to be sequestration in P-bodies, so the cell can use it for later. Whereas in siRNA, you're just going to have the piece of DNA or the target DNA cleaved and degraded. Yeah, next slide.
I just wanted to add that, yeah, she does, at least when I took the course, she did ask like the difference between these 2 or similarities between these 2 pathways. But yeah, it's just like Saeed said, that the miRNA pathway is endogenous, so it's like within our genome, whereas the siRNA pathway is exogenous. So that materials of the double-stranded RNA can be inserted into a person.
Yeah.
Yeah.
And also the thing with that is that that's why it will be used for lab purposes, which is just like another thing that you asked about.
Yeah, so does anyone have any questions at all on the RNAi stuff? I can't see the chat.
Sophia, your question. Yeah, so the end result, there in miRNA, you can have either of the 2. You can either have the mRNA degraded or sequestered, but mostly you're just going to see it being sequestered. But in siRNA pathways, you're just going to see the target DNA being degraded. You're not going to see any sequestration of that. So yeah, hopefully that answers the question. Yeah. All right, so go to the next slide. So another point why RISC can't just grab a single strand of RNA, you think that would just be more efficient for the cell to not have to go through all the processing by Deisser, or by Drosha and Deisser. And basically the reason that all you need to know for this class is that single strands of RNA are a lot more unstable than your double strand. Whereas your double strand RNAs are going to have more protection inside the cell. If there's just one strand of it, then it's more likely going to be marked for ubiquitination by the cell. So basically that's the main reason. Your double strand RNA is going to be able to last longer in the cell to where it can need to be used by the RISC complex. And the other thing is that single-stranded RNA can't really work with the RNAi pathway. RISC requires the 2 strands. And if you think about that, that just allows it to be able to pick one of the 2 strands for being used as a guide. And yeah, the other last thing to know is that RNA can also form other higher order structures by itself, right? Usually we think of the amino acid or proteins folding on themselves, but RNAs can do that too, and you can see that in the stem loop hairpin structure of your PRI microRNAs, which, if you look back in the pathway earlier, those hairpin structures are what Drosha is going to use to cleave your pre-microRNAs into their smaller pieces so that it can be then exported out of the nucleus. So if we have any questions on any of that.
I have one more question.
Yeah, sure.
So what causes, like, say in the case of like miRNA, like causes it to initially hairpin before it's like processed by Drosha and stuff?
Oh, I actually don't know why it forms a hairpin. I do know that Drosha is going to use that hairpin as a substrate to be able to then cleave it, but I don't know why the hairpin forms like in the first place.
I don't think she ever will ask you why, or I think that's like more beyond the scope of this class. But it's just that it's just something to keep in mind that it has a hairpin, but we don't know why it does.
Okay, thank you.
Yeah, sure.
Are there any other questions about RNAi in the chat or anything? Okay. All right, so since there aren't any more questions on RNAi, I'm going to jump into cell signaling. So this is just an overview of the signaling stuff before I'll get into the pathways. So yeah, so this is intro to cell signaling. And we learned in class that cells have signals that tell them what to do. And instead of having just a single signal, cells actually will have multiple signals. And so a cell is able to have multiple signals at once. As you can see in the image, the cell has the surviving signal, the grow and divide signal, differentiating signals. You can see with the A, B, C, D, the letters, that there's multiple signals happening. And this is because cells need signals to survive, and it's pretty difficult to keep human cells alive in labs because you're basically removing them out of a human and putting them into a petri dish where they don't have all those signals needed to survive. And yeah, signals can be small molecules or ligands, which are proteins, which can be proteins, but there are some signals that aren't proteins. And signals can be received by extracellular receptors, so receptor on the outer surface of a cell, or intracellular receptor, which is on the inside of the cell. And yeah, so another important thing from this slide is that if the cell does not have survival signals, then the cell will die, which you can see in the last image. That cell doesn't receive any signals, so it becomes an apoptotic cell. So yeah, that's signals. Does anyone have any questions on this slide? Okay, I'll keep going. And just like I said, signals can either go on cell surface receptors, or they have receptors that are located inside the cell. And so signals are small molecules or can sometimes be proteins. And they're hydrophobic signals and hydrophilic signals. The hydrophobic ones are able to pass through the cell membrane and bind to receptors in the cytosol. And this is because the cell membrane, at least the human ones, are pretty selective. So I think they're just called semi-permeable membranes. So they're selective in terms of what they let pass through the membrane. And so this is why hydrophobic signals can pass through. And some examples of these are cortisol and nitric oxide and some of the sex hormones. And yeah, so nitric oxide is considered to be hydrophobic, even though it has the polar groups you can see there, nitrogen and oxygen, because both of those two atoms are highly electronegative, so they're able to cancel each other out. So that would make nitric oxide hydrophobic. So that's why that one can pass through. I just thought I'd clarify that one because at least when I took the course, there was a bit of confusion whether that was hydrophobic or hydrophilic. And then we also have hydrophilic signals, and these ones are not able to pass through the membrane because these signals are polar or hydrophilic. So they need to bind to the cell surface receptors. So these are things like growth factor signal and insulin. And for the sake of this class, we're just going to look at cell surface receptors. So these are the hydrophilic signals. Does anybody have any questions on this slide? Okay. And so with signaling, you generally have two main outcomes that can happen. you'll either have a change in protein function or you'll have change in protein levels and expression. And so both of these processes are highly regulated. And the general idea for this class is that it's much faster to alter a protein's function than it is to change protein levels and expression. And you can see here in this diagram that if we were to alter protein expression, then you have to have a signal binding to the receptor, and then that leads to a whole signaling cascade, which results in the last component of the pathway activating a transcription factor. Then a transcription factor enters the nucleus, and then it will interact with the DNA, and then transcription occurs, and then you have to have translation, and then all of that stuff has to happen, which takes a lot longer than altering a protein's function, which is just like changing the job of a protein. So yeah, does anybody have any questions on this slide? Okay, I'm gonna keep going. We also looked at how sometimes we can have signals that are exactly the same and bind to similar receptors, but they can have different responses or outcomes. So this depends on the cell type. So if you have different cell types, then the signal may have different effects in the cell. And this is because different cells have different proteins, and there are different downstream responses within the proteins. And so an example I put of this is trying to think of all the things that would happen when you're feeling stressed out, which I'm sure might be applicable to some of you with the finals week. So when you're stressed, you have several different things that happen, but it's mostly regulated by the adrenaline pathway. So the adrenaline pathway is much like a signal transduction pathway. So you have like the epinephrine ligand binds, and then you have like activation of downstream components. And so all of that stuff will result in different things, such as your heart rate increasing, your digestion slows down, you begin to like profusely sweat, and then you have saliva accumulating. So you have all these different responses happening. And that would make sense because sometimes when you're stressed or if you're in like a fight or flight situation, you want different things to happen within the body so that you're prepared to face whatever you're facing. So that's why you have a similar signal causing different outcomes to happen within different cells. And so yeah, does anybody have any questions on this slide here? There's nothing in the chat, Saeed, because I just can't see the chat.
No, I don't see anything.
Okay, cool. Yeah, so this is just more about receptors. And I believe I said most of these things. So I guess, yeah, this is pretty much a summary of what I just said. But you have the extracellular receptors and then the intracellular receptors. And a good thing to know is that the receptors that are intracellular will be able to accommodate signal or ligand molecules that are small and hydrophobic. So that's how they're able to like sneak through the cell membrane. Then your extracellular signals are usually large and hydrophilic. So yeah, you can see in the picture on the right how the one on the outside is a lot bigger and bulkier. So it has to be on the outside on the cell surface receptor, whereas the signal inside the cell and the cytosol is a lot smaller in shape and hydrophobic. So yeah, I'm going to just keep going with this. There's no questions. And okay, so then the next section that we talked about in terms of cell signaling stuff is signal amplification and crosstalk. So signal amplification describes how signals can be amplified if there are enzymes in multiple steps in a transduction cascade. And so for this, I tend to just think of like sort of a pyramid shape. So the top of the pyramid, you have like the peak of the pyramid, and you have so you have just like one thing that happens, and then it leads to a cascade of just a bunch of signaling as you go down the pyramid, so it gets larger. So in these diagrams here, we have so in the one on the right, you have one epinephrine signaling molecule that will bind to the receptor. And so that one epinephrine molecule eventually causes 20 cyclic AMP molecules to be produced. And then each of those cyclic AMP molecules can bind to pKa, which is protein kinase A. And then every individual protein kinase A can go on to activate 3 phosphorylated proteins. So you can see how just having one molecule, one signaling molecule, can cause all these things to happen. And it results in just a large production of downstream components and a large amplified or escalated cell response. So does anybody have any questions on this slide? Okay. And yeah, I just thought I'd put a few more diagrams on signal amplification because when I took the course, there was some confusion with signal amp and crosstalk, but they're 2 entirely different things. So you can see in the diagram what I was saying earlier about the pyramid scheme, how you just have one signaling molecule leading to just a bunch of things happening at the end of the pathway. And so, yeah, so you just have one molecule and then leads to production of adenylyl cyclase and cyclic AMP, and then cyclic AMP binds to protein kinases, and all those protein kinases cause multiple target proteins to be activated. And yeah, this is just a diagram on signal amp. So if there are no questions, I'm going to go ahead and continue to crosstalk. So crosstalk describes when you have components of one pathway affecting another. And so this is when you have proteins that from different pathways communicating with each other to get a signal across. So you can see in the diagram, we have 2 pathways, pathway A in yellow, pathway B in pink. And so where the 2 sort of converge, you have a protein that is part of both pathways that's able to communicate to both pathways. So effectively, the protein that is part of both pathways can cause one pathway to shut down or be upregulated, depending on certain things. So yeah, this is just a diagram that I found of the crosstalk. So does anybody have any questions on this slide? Okay, I'm gonna keep going. And yeah, I just found more diagrams of crosstalk because I just felt like the ones I showed in the other review just wasn't as clear. So yeah, you can see how there's multiple pathways. At least in the one on the left, you have 3 different colors that represent 3 different pathways. And you can see how one pathway can have a component that can inhibit or upregulate another pathway. And so that's just an example of crosstalk. And I also I have an analogy for crosstalk, which for the sake of time today, I won't go through it again, but it's part of the YouTube channel. I gave an analogy that has to do with real life and having like groups of people getting a message across. So I think it'll be good for you guys to check that out if you have time. Yeah, so this is crosstalk. Does anyone have any questions about this? Okay. Next, I'm going to talk about kinases and RTKs before we dive into the pathways. So this is just a quick summary of protein kinases, what they are. And so some of you on the second exam seem to have confusion with what are enzymes and what are not enzymes, and whether kinases can be enzymes in proteins. But just know that for this class, that kinases are enzymes, and they're a special kind of enzyme that catalyze phosphorylation. And kinases are enzymes, and enzymes are proteins. So yeah, enzymes are proteins that catalyze reactions, and kinase is just like a special category of enzymes. And kinases are able to cleave phosphate groups from ATP and attach it to a substrate. And so you can see in the diagram It's not always shown in your pathway, so you don't usually see it for the pathways we studied, at least in the diagrams, but just know that in the background, when a kinase phosphorylates something, it's actually removing a phosphate group off of ATP. It's cleaving that phosphate group, and then it's tacking it onto that target protein. So it's just a good thing to keep in mind. But it's not always explicitly drawn in images, but just know that that's what kinases are doing. They're going through a reaction. Yeah, so that's pretty much the overview of the protein kinases before I talk about RTKs. Does anybody have any questions on this? Okay, I'm gonna keep going. This is just a quick recap of what RTKs are before I get into the pathways. So RTKs are essentially monomers that have half of it sticks out outside the cell, so half of it's extracellular, and then it has an intracellular portion which contains the tyrosine kinase domain. And just like I said in the other slide, kinases are enzymes that catalyze phosphorylation. And majority of the enzymes we deal with in this class are proteins. And RTKs, how they work is that a signal comes along and it will cause the two RTKs to dimerize. And then once the RTKs come close together, the dimerization occurs. And then this interaction will stimulate the kinase activity. And this is what causes the RTK monomers to autophosphorylate each other, or those tyrosine kinase domains to interact because they're close together. They're able to cross-phosphorylate each other. And then once the RTKs monomers cross-phosphorylate each other, they become active. And activation produces binding sites for intracellular signaling proteins. So does anybody have a question on this slide? Okay, I will keep going. So yeah, there are 4 pathways that you'll have to know for this class, and we're just gonna roll through all of them. But this is just essentially the 4 that we're gonna look at. So first pathway we talked about was the RAS, MAPK, RTK pathway. Sorry. And just like I mentioned earlier, the RTKs is what begins this pathway. So you have the signal that binds, the RTKs dimerize, and the intracellular kinase domains become active, and they will cross-phosphorylate each other because they're in close proximity. And then the autophosphorylation creates binding sites for other proteins. And then the binding sites that are on the active RTK is a binding site for the adaptor protein, which is also called GRB2. So your adaptor protein will bind to the phosphorylated tyrosine on the active RTK, and that is called the SH2 domain, by the way. So wherever you see a phosphorylated tyrosine, that binding site area is called the SH2 domain. So once the adaptor protein, or GRB2, binds to the SH2 domain on the active RTK, this will cause GRB2 to recruit the RAS activating protein, which is the light blue protein. And so once this occurs, the RAS activating protein will exchange GDP for GTP in RAS. And a way you could word that, if you're looking for how to write that, is you could say that the RAS activating protein causes RAS to have lower affinity for GDP and higher affinity for GTP, because she does mention how GTP is available at a higher concentration within the cell. So once RAS loses affinity for GDP, then there's enough GTP available in the cell for it to diffuse into RAS's binding pocket. So once GTP is attached to RAS, RAS is now in its active state. And so it is able to go onwards within this pathway and activate the MAPKs. Oh, I'm sorry. Does anybody have any questions so far on anything I explained for this pathway? Okay, I'm just gonna keep going. So now we have activated RAS protein, and it's going to go and interact with the MAP kinase. So RAS, once it's active, will interact with MAP kinase 3, which Professor Francis calls MAP kinase, but you can abbreviate it to MAPK3. You don't want to write kinase so many times. So yeah, so RAS interacts with MAPK3, and then MAPK3 will phosphorylate MAPK2 at 2 different positions, hence the 2 yellow P's on the diagram. And then the MAPK2 will phosphorylate MAPK1 at 2 different positions. And then your MAPK1 will go onwards to phosphorylate other proteins. And it also has the ability to activate transcription factors, and those transcription factors at the bottom right of the screen can enter the nucleus and cause changes in gene expression by binding to DNA. And just a real quick thing to highlight for this pathway is that RAS is not a kinase, so that's why you don't see a phosphate next to the MAPK3. So we don't actually know how RAS activates MAPK3. We just know that it does activate MAPK3. So it would be incorrect to say that RAS phosphorylates MAPK3, when in reality it just causes MAPK3 to become active. So just make sure to keep that in mind if you're writing a response for this question, or if you're trying to figure out how to explain this pathway. So RAS, again, RAS does not phosphorylate MAPK3, it just will activate it. Yeah, so does anybody have any questions at all on the RTK RAS MAPK pathway? Saeed, there's nothing in the chat?
Oh yeah, and there's nothing.
Okay. So yeah, so for the slides for the pathways, I broke it up into smaller chunks. And then I also just have the entire pathway laid out in one slide, just so you guys can go back and practice. So if you can sort of explain what's happening and the steps in the normal pathway, then I'd say you're in pretty good shape in terms of explaining what would happen if something went wrong, because that's something that Professor Francis likes to ask a lot on the exams. So like what would happen if you broke something in the pathway, or if you had a mutation in a certain protein, or you did something to a certain element in the pathway. Those are the types of questions you would expect to see for this exam in terms of regarding the pathways. So yeah, so that concludes the RTK RAS MAPK pathway, and highly encourage you guys to use this blank slide here with no words and just a picture just to practice it. And yeah, so I'm gonna go on and explain the next pathway. So the next pathway we talked about was the survival pathway. And these questions were just questions I had in there to keep in mind about the pathway. But basically all it is just, can you explain how it works? And then like if you're able to cause cells in this pathway to die, because the whole point of this pathway is to cause survival to occur. So if you mess around or tweak any components of the pathway, how could you do it in a way to cause the cell to die instead of survive, even if the survival signal was present? So those are just things to keep in mind about this pathway. And so for the survival pathway, it starts with the RTKs. And so the survival signal arrives in the form of a dimer, and then the RTKs will dimerize and phosphorylate each other. The tyrosine kinase domains phosphorylate each other. And then in this pathway, you'll have PI3 kinase will actually bind to the SH2 domain on the active RTK. And then once this occurs, PI3 kinase will phosphorylate inositol phospholipid. And inositol phospholipid is anchored to the cell membrane. And so once PI3 kinase activates inositol phospholipid, it will cause inositol phospholipid to, sorry, I'm just thinking out here, sorry. So once PI3 kinase phosphorylates inositol phospholipid, then that will cause inositol phospholipid to go from diphosphorylated to triphosphorylated. And so once inositol phospholipid is in its triphosphorylated state, then that means it's in its active state and it becomes a binding site for protein kinase 1, as well as AKT. And so, yeah, so this step is just the RTK and then helping to make inositol phospholipid in its active state so that it can go onwards to do its job in the pathway. And yeah, something I forgot to mention was that inositol phospholipid is not a protein, which is something that's, I guess, new to us with the pathways, because normally most of the things are proteins, but yeah, it's not a protein, it's a phospholipid. So yeah, does anybody have any questions at this point? Okay, I'm gonna keep going onwards with the pathway. So once we have our phosphorylated inositol phospholipid, or it's in its active state, then it will bind to AKT, and wherever the PIP, the phosphorylated inositol phospholipid, binds to, that is called a pH domain. So you can see here that protein kinase 1 and AKT both have pH domains. So anyway, so the PIP will bind to AKT in the pH domain. And then in order for AKT to become active, it needs to be phosphorylated by 2 other kinases. And so you see how we have the brown protein kinase 1, and then the purple kinase. So what happens here is that you have to have both of them activating AKT in order for AKT to become active. So protein kinase 1 will come along and phosphorylate AKT. If you can see it on the left side. And then once that occurs, AKT will undergo a conformational shift. And so that actually exposes the binding site for protein kinase 2, which is the purple protein, to come along and bind to the binding site on AKT. So yeah, so there's subsequent phosphorylation, so one after the other, of protein kinase 1 and protein kinase 2. And so once AKT is activated by both of these, once AKT is phosphorylated by both kinase 1 and kinase 2, then AKT is in its active state. And so AKT will dissociate from the protein kinase 1 and protein kinase 2, and it will go onwards to promote cell survival. And so it will do that by entering the cytosol and binding to BAD, which is the next part of the pathway, which I'll get to. And just something in here that I put on the slides was that I'm not sure she asked you about it on a problem set or if she asked in the exam, but she mentions that there is this phosphatase called PTEN, and it has the ability to remove phosphates off of PIP3, so the phosphorylated inositol phospholipid. So that's just a phosphatase that can remove phosphates. So I just thought it was something good to know. Does anyone have any questions on the steps in this pathway so far? Okay, so now we have activated AKT, and it goes onwards to do its job. So activated AKT will dissociate from the kinases and the inositol phospholipid and enter the cytosol. And then from there, the AKT will bind to BAD. And when this happens, when the active AKT binds to BAD, it will actually inactivate BAD. So this is a point of confusion for a lot of people, because up till this point, you've learned that when you phosphorylate something, you make it active. But this is actually a different case with BAD, because when you phosphorylate BAD, you're making it inactive. So when you phosphorylate, when active AKT goes and binds to BAD and phosphorylates it, it makes it inactive. And so this is a good thing, because when BAD is normally active, it is bound to BCL2. And so BCL2 is an important protein in this pathway because it is the apoptosis inhibitor protein. And that essentially is just a fancy way of saying that it can prevent cell death. It's the protein that prevents cell death. So normally when BAD is active and it's bound to the protein that prevents cell death, then it is preventing that protein from going onwards to save the cell from dying. So when BAD is active, it's a bad thing because it's just holding the protein that's supposed to save the cell hostage, so it's unable to do its job. So that's why AKT will phosphorylate BAD, which inactivates BAD, and then that leaves BCL2 free to prevent programmed cell death. And BCL2 will roam free, and it will prevent programmed cell death. I think by binding to mitochondria, but I don't think you have to know exactly how BCL2 prevents cell death. Just know that it is important in preventing cell death. And so yeah, so that's pretty much the survival pathway. And I am gonna talk a bit more about BAD because I feel like that's a confusing part. So just like I said, apoptosis is cell death, and BAD is a protein that promotes cell death or causes it to happen. And then BCL2 is a protein that inhibits cell death. And so when you have active BAD, it's bound to BCL2. And when BCL2 is bound to BAD, it's inactive. And so this causes cell death. But if AKT comes along and phosphorylates BAD, that makes BAD inactive. And so when BAD is inactive, it can't bind to BCL2. And then that means BCL2 is active and it's able to it's able to roam freely in the cell and inhibit cell death. And so that means that the cell would live. Does anybody have any questions on the survival pathway so far? Okay. And so yeah, I just have a visual explaining the two scenarios that I just said. So you can see when you have active BAD, have cell death. And then when you have inactive BAD, you have cell survival. So yeah, that's just the two scenarios. And this is just, again, a diagram of the survival signaling pathway, just for you guys to practice on your own and see if you can explain what's going on here. And just like I said, the idea is that if you can explain what normally occurs in the pathway, if you understand why things occur in the pathway, then you'll be able to figure out what happens when you break the pathway or mutate it or manipulate it. Okay. So next pathway I'm going to talk about is the JAK-STAT pathway. Questions to keep in mind about the JAK-STAT pathway, which I had from the other review that I did, was explaining how it works, and then is JAK-STAT considered to be an RTK or not? And I'm going to skip over this slide because I just went through what RTKs are. But yeah, so JAK-STAT pathway is involved in multiple processes. So in this class, we looked at the production of milk genes, which is done by the ligand prolactin. And so the JAK receptor is not considered to be an RTK, even though it's a receptor, but it's not an RTK because it does not have a kinase domain. And you know that RTK stands for receptor tyrosine kinase, so it would not fit the definition. So what makes the JAK receptor different is that it has kinase subunits that associate with it. So yeah, so it's not a receptor tyrosine kinase, and it's because in this pathway, it's not the receptor monomers that cross-phosphorylate each other, but it's just the JAK proteins that happen to be attached to the receptor that cross-phosphorylate each other. And then once the JAK proteins cross-phosphorylate each other, then they're able to phosphorylate the receptor. And yeah, so JAK stands for just another kinase. and I think it took on the new name Janus kinase. So from the name JAK, you can tell that a JAK is an enzyme, because enzymes are kinases, just like I said. And yeah, so JAKs have receptors, but they're not. Yeah, so there's receptors in this pathway, but they're not RTKs, and the JAK receptor is not an RTK. Yeah. Does anybody have any questions on what I just said so far for this slide? Okay. So now I'm just going to explain how the pathway works. So you have the JAK proteins in light blue are bound to the receptor. But before that, the very first thing that occurs is your signal once again will come and bind to the receptors, causes the receptors to dimerize. Then once this happens, the receptors dimerize and they get into close proximity with each other, so they'll touch each other. And then once this happens, the JAK proteins, the light blue proteins, are able to cross-phosphorylate each other, and then they become active. And then after that occurs, the JAK proteins themselves will phosphorylate the receptor. And then after the receptor is phosphorylated, then it will create binding sites, which again, they're SH2 domains. They'll create binding sites for the STAT5, monomers to come along and bind to. So you can see that the peachy colored or light pink salmon colored proteins on each receptor will come along and bind to that SH2 domain on the active receptor. And so once this occurs, the STATs become phosphorylated by the JAKs. And then once that occurs, the STATs will dissociate from the receptor and they will connect with each other. So they will bind to each other because the STAT5s have SH2 domains within themselves. So each STAT5 protein will attach to the other protein and make a STAT5 dimer. And then once that occurs, the STAT5 dimer is able to enter the nucleus and it can act as a transcription factor. And the way that they're able to enter the nucleus is through important, which you know important from the nuclear import and export. And yeah, I think that's pretty much it for the JAK-STAT pathway. So does anybody have any questions? And this is just a diagram of the JAK-STAT pathway. So you can walk through and practice explaining how things work in here. And yeah, so that's it for the JAK-STAT pathway.
Okay, now we're going to be going over gene protein coupled reaction, or GPCR pathways. And so basically, if all you're going to need to know is that, can you explain the different steps of this pathway? So could you explain to someone who has never taken this class before but has like some knowledge of biology and biochemistry? And another important thing you're going to want to know is that make note of the Jeff analog in this pathway and the GAP analog, because it's not going to have those, but it's going to have other machinery that's going to act with the same function or purpose in mind. So next slide. Sorry, could you go back to? Yeah. So basically, we're just going to walk through the different steps. So it's in the name. You're going to have your G protein, and then you're going to have its receptor. The receptor, which is going to be a 7-transmembrane bound receptor, and that one, it's in its default state, it's going to be bound to the G protein, which is going to be made up of a heterotrimer. So basically, it's going to have 3 subunits. It's going to have your alpha, beta, and gamma subunits. Gamma, which is going to be bound, as you can see in the diagram. It's going to be bound to the membrane by itself. So basically, what's going to happen is that in its default or off state, it's going to be the receptor is going to be bound to GDP, but it's when a ligand comes in to activate it, GPT is going to be diffused out, and that's going to make it more likely for GTP to come in, which is then going to start off the pathway. So um, when GTP comes in, then G protein is activated. It disassociates, the alpha portion disassociates from beta and gamma, which, as we mentioned before, the gamma part is going to be um, separately attached to the membrane. So those two pieces, gamma and beta, are going to go off and be used in other pathways. And then for this pathway, you're going to want to watch the alpha subunit, which is then going to go out um, and then be able to be used from different pathways because it's going to be in its active state. And the way that the pathway is going to turn that off is that RGS is going to come in, and it's basically, and it's going to attach to, as you can see here in the diagram, it's going to attach to that alpha subunit. And when that happens, it's that an important distinction you're going to want to know is that here is where RGS is just going to accelerate at the alpha subunit's already like intrinsic ability to hydrolyze GTP. So basically, the alpha subunit this whole time that's active, it's going to be hydrolyzing is GTP. It's just going to happen naturally at a very like slow rate. So RGS is going to come in, and it's just going to accelerate that to where the GPT is going to more quickly be hydrolyzed to GTP, which is going to deactivate it, which is going to then cause the alpha subunit's effector to disassociate. And with that gone, then the protein complex is going to come back together where the alpha subunit is then going to join again with the 7 transmembrane and the 2 beta and gamma subunits. So if there's any questions on that, otherwise we can go on. Oh, and another important portion is that you should know that the G protein, the complex itself, is going to be your Jeff analog, right? Because that's going to be losing the GDB to then infusing the GTP to be active. And then RGS in this scenario is going to be your gap analog, which basically just means it's not an enzyme itself, it just accelerates its substrate's intrinsic ability to hydrolyze. So there's no questions on that. We can go to the next slide. So basically this is just going to recount the steps that I just talked about. Just know that if you know these 6 steps, then you're pretty much golden. That, and along with knowing that the G protein coupled receptor, the whole thing is going to be your Jeff, or the receptor is going to be your Jeff analog, and again RGS is going to be the gap one. So yeah, as long as you can memorize these steps, figure out, okay, so if say step 3 doesn't work right, then what's going to happen? Then you should be set for the exam.
I wouldn't say memorize. I just more say like having a general understanding, like when you look at the diagram of what's going on in the pathway, what happens.
Yeah.
Yeah.
Yeah, that's a better way to say it.
And then that way you can figure out that, hey, what happens if there's a loss of function mutation in RGS, or what happens if you know, the ligand didn't bind to the GPCR receptor, what events would be affected, what would stay the same, stuff like that.
Yeah, basically any like say loss of function or even gain of function, any way, like any points along the steps. If you know what would happen then you should be good.
Cool.
So if you go to the next. Yeah, so some common misconceptions. Just gonna recount what we kind of mentioned earlier. GAP and Jeff functions are not enzymes themselves. They help proteins or their substrates of the pathway. So that's like a small but like important distinction to make. Again, is that those themselves, like your RGS or your G protein or whatever have you, those aren't going to be enzymes, right? They're not going to catalyze anything. They're just going to be assisting. Again, Jeff is going to be promoting GDP to dissociate, which is going to allow a higher probability of GTP to dissociate into the protein. Right, it's going to make it more affinity, have a higher affinity for the GTP. And then GAP, its function is just to bind to the GTPase. Again, if we just use the alpha subunit as an example, that would be your GTPase in that scenario. and its gap is just going to accelerate its own intrinsic enzymatic activity for GTP hydrolysis. But yeah, and I know the last thing, GRAB2, it binds to phosphorylated tyrosine on the RTK because of its SH2 domain, and then it also recruits the RAS activating protein on the SH3 domain. So that's an important distinction between the SH2 and the SH3. So yeah, and we have any questions on that?
Cool. So I think that's it for pathways, and then this is the very last section of today.
Mhm.
So almost done, guys.
Yeah, so just after this, we should be all set. Yeah, microtubules. So if you go to the next slide, you have three types. So microtubules are, as you know, like just another form of the cytoskeleton. Later on, obviously, in the later reviews, we're going to go over the other two, but just to mention them here, we have actin filaments, which are basically going to help with muscle contraction and cell motility. Right, it's going to help with cytokinesis, vesicle organelle movement, and it's also going to maintain the overall shape of the cell. And then you have the intermediate filaments, and these are just going to also help with cell structure. They're going to bear tension that the cell might undergo, and it's also just going to help with the shape. And also, it's going to be anchoring the nucleus and other organelles in place inside the cell. And then for this topic, or for this section, microtubules, these are what's going to help the cell resist compression. It's also going to help with vesicle movement, and the important part is that it's going to be used during anaphase, when replicated chromosomes are going to be pulled to the opposite side of the dividing cell. And other than that, it's also going to be used in structural elements of the cell, such as, say, if a cell has a flagella or cilia or centrioles, those are going to be made up of microtubules. So on to the next slide, we're going to have our spindle assembly. So right, this is going to happen during your anaphase of the cell. Microtubules are going to be forming the spindle, and they're going to separate chromosomes during the M phase by attaching to the kinetochores on the chromosomes. So we have three different types of microtubules, right? We have your green aster microtubules, your kinetochores, and then your interpolar. To start off, we have we have the aster microtubules. So those are going to be growing, they all grow from the centrosome, but your aster microtubules are going to be attaching to the cell cortex, and that's just another name for the inner layer of this plasma membrane, so the inside of the cell. And then you have your kinetochore microtubules. These are going to be growing from the centrosome and then attached to the kinetochore on the chromosome itself, right? So that's what's going to allow the cell to actually pull the physical chromosomes apart. And then we have your interpolar microtubules, right? These are going to also grow from the centrosome, but they're going to overlap. You can see here at the top of the spindle, right? So those are going to grow and shrink, which are basically going to, at the final stage of mitosis, are going to pull apart the actual spindle poles and the cytoplasm to the other side, or to each polar side of the cell. If we're good there, we can go to the next slide, which is on tubulin, right? So these are going to be your dimer monomers of the microtubules, right? So these are going to be the pieces that make up the microtubule itself, and they're both going to be GTPases, right? So again, they're going to have the ability to hydrolyze GTP to GDP. The only difference is that the two subunits, your alpha and your beta, alpha's going to have, well, they're both going to have the GTP binding pocket, but in the alpha side, right, because it's on the minus end, that pocket's going to be blocked because that's where the beta portion is going to be. So alpha can't hydrolyze, or it can't grow GTP on that side as fast because that pocket's going to be blocked. So just by virtue of that, that's going to be your minus end. And that just means that you're not going to be seeing as much growth there as opposed to the beta side, which is going to be your plus end, right? On this end, the GTP pocket's going to be open, which means basically it's that GTP is going to be allowed to come in and be hydrolyzed, or regulated or have GTP come in there. So that'll allow it for to grow on that side. So if you go to the next slide, we're going to look at the structure of microtubule, right? So it's going to be made up of those heterodimers, right? The alpha and beta tubulin. Again, they're going to be polar molecules, the heterodimers are, so they're going to have one end, the minus end, where you're not going to see as much growth as the positive or plus end. It's important to note that it can grow on either side. It's just going to be more likely or faster to happen on that beta or plus end. So you're going to see that microtubule is going to be a combination of 13 protofilaments, which are basically just going to wrap around the circumference of the tube, which again is going to be hollow. You're just going to have lumen or space in between, as you can see in the diagram. And you're going to have the negative end or the alpha subunit in cells in vivo going to be embedded into a microtubule organizing center, or MTOC, which we're going to talk about later, but that's where the base or the minus end grows from. And then the other side, the plus end, is going to extend forward basically by virtue of tubulin assembly on that side. So if you go to the next slide, we'll talk about microtubule nucleation. So basically this is just the idea of gamma tubulin rings being formed, which are going to act as a base for the start of microtubule growth, right? So because of that, you're not going to need microtubule to just grow randomly or from scratch inside the cell. You're going to have a base for it, which is going to be your gamma tubulin ring, which comes in either the SC or RC. And that just means your small complex assembly or your ring complex. And here you see that happening in vitro on the right, or in vivo on the left, because of the microtubule complex that we mentioned earlier. So first find there, we're going to go on to dynamic. Give me a second. Oh yeah, so dynamic instability. So basically, this is just the main idea that microtubules can grow and shrink independently of one another, right? Just because you have, say, a petri dish full of microtubules, some are going to be growing and some are going to be shrinking, and they can change that at any given time. And that's basically just dynamic instability. So you're going to look at whether the rate of any given microtubule is going to be growing. So basically, you're going to see more. You're going to see more tubulin addition on the plus end as opposed to hydrolysis, or vice versa. So, right, if you have hydrolysis happening at a greater rate, basically, which means that GTP is going to be turned into GDP, you're going to see the microtubule become depolymerized or unstable and start to shrink. And then you can see that here on the right side, you can see there's no GTP cap because the tubulin is going to be hydrolyzed faster than it can grow, and that's just going to lead it to shrink. Whereas on the opposite side, if the opposite's true, you can see that tubulin's coming in with bound GTP, and since that side is happening faster than the other negative side and can hydrolyze it, you're going to see that formation of a GTP cap, which just means that the microtubule is going to be growing. So if we're fine on that, we can go to the next slide, which is going to be focused on, as we mentioned before, the microtubule organizing center. So you're going to find these in vivo or inside the cell, and they're just going to form as like a jump start for the microtubule, right? It's going to be a base for that minus n to grow out of, or that minus n is going to be embedded inside of the MTOC, and then the growth is going to be on the opposite side, or the plus side happening. And the other important thing to note here is that in cilia and flagella, these are where you're going to see the organizing centers a lot. Right, and that makes sense because you're going to want to see movement in both of those structures. And the other thing is that centrioles in our cells are just vestigial structures, and they probably don't play as much of a role in microtobial assembly, as opposed to centromeres, which contain that gamma tubulin ring that we talked about before. So if we're fine there, we can go to the next slide on, again, just growth and shrinkage, right? Growth is just going to be when tubulin's coming in with GTP bound faster than it's being hydrolyzed on the other side, right? You're going to have that beta tubulin and GTP cause for growth. But on the opposite side of that, if it's being hydrolyzed faster, then the beta and the GTP can come in, then it's going to be unstable and it's going to cause it to shrink. Next slide. Basically, in vivo or in the cell, you're only going to see growth and shrinkage on the plus side, right? Because that minus end is going to be embedded. It's going to be blocked off from any tubulin coming in because it's embedded inside of the organizing center. And that is in contrast to the next slide, where microtubule growth is seen in vitro or in the lab. Here you can see addition and depolymerization happening at either side of the microtubule because both ends are exposed. But you're just going to want to note that that rate is going to be different, or it's going to be growing, or it's going to be adding faster on the plus end as opposed to the minus end, even though it's happening on both sides. Yeah, and so basically the other thing you want to note is that in vitro, you're going to have some key factors that you're not going to see inside the cell. And those are basically just going to be, you're going to be limited by the number of tubulin and GTP available, right? So whenever those run out, then you can see a switch from growth to shrinkage. And yeah, that's pretty much that for microtubules. If you guys have any questions.